Let's take a closer look at how genes are regulated in bacteria. Genes in the operon encode proteins that allow the bacteria to use lactose as an energy source. First, we see RNA polymerase bind into the promoter. The promoter is a binding site for RNA polymerase, the enzyme that performs transcription. Next, we see the LAC-I gene. The LAC-I promoter is going to allow for transcription to occur, where RNA polymerase will transcribe the LAC-I gene and the repressor protein will be made, represented here by the monkey. The LAC repressor will act as a lactose sensor. It normally blocks transcription of the operon but stops acting as a repressor when lactose is present. The LAC repressor senses lactose indirectly through its isomer allolactose. Allolactose is represented here by the other monkey. Next, we move on to the LAC promoter and the LAC operator. The LAC operator is a negative regulatory site bound by the LAC repressor protein. The operator overlaps with the promoter, and when the LAC repressor is bound, RNA polymerase cannot bind to the promoter and start transcription. Here we then see that the LAC operon contains 3 genes, also called structural genes, the LAC-Z, the LAC-Y, and the LAC-A. Each of these genes are transcribed as a single mRNA under control of one promoter, which is here. When there is no lactose, the LAC repressor binds tightly to the operator, preventing transcription by RNA polymerase. So here's RNA polymerase. It's unable to get by because the monkey is blocking its way, the repressor protein here. Therefore, no transcription of the structural genes Z, Y, and A. When lactose is present, the LAC repressor will lose its ability to bind DNA. It will float off the operator, clearing the way for RNA polymerase to transcribe each of the structural genes. Allolactose binds to the LAC repressor and makes it change shape so it can no longer bind DNA. Allolactose is an example of an inducer, a small molecule that triggers expression of a gene or operon. The LAC operon is considered an inducible operon because it is usually turned off, repressed, but can be turned on in the presence of the inducer allolactose. Carissa is a young new mother who has to get up in the morning at 4 a.m. to get ready for work. Her 5-year-old daughter is scared of the dark and wakes her mom up during the night. Carissa's daughter waking her up during the night is the repressor. Carissa getting a good night's sleep represents transcription. In order to keep her daughter asleep during the whole night, we need an inducer. Using a night light in the room will work. This 5-year-old will have a higher affinity for the inducer than for waking her mom up. This regulation type is therefore inducible, where it's negative because it represents a repressor, and inducible because we have an inducer, the night light.